Okay, so if you recall yesterday, we did this. Let's listen. All right, we talked about asexual reproduction. We're going to transition to talking about DNA. And if you are one fertilized egg, that means you're one cell, two genes from two different sources fused together. And that's asexual reproduction, or sexual reproduction. What is it? That's sexual reproduction. Now, up on the screen I have asexual, because the moment you are that fertilized egg, for the rest of your life, you're going to mass produce your cells or replace the cells that once you're fully formed. And that's asexual reproduction. We've been talking about it, calling it mitosis. But what I want you to understand is you have all the genes you'll ever have at the point you're one cell. And from that point on. That make sense? Yeah. It seems kind of, for me and Mr. Martini, that's something like, when we were on your side of the desk, we didn't really fully understand that. Right? We understood like embryonic development. That's when you start as an egg, fertilized egg, and you grow fetus, and you're eventually born. If we're talking about a human, all right? But so what I want you to start thinking about genes and how they turn on and off. So first, we just to review. Remember, we're human. We have 46 chromosomes. Different organisms have different number of chromosomes. They're always in pairs because you get one from one parent and the other from the other parent. That's how you get your 23 from your mother, 23 from your father. All right? That's how you have your 46 chromosomes. Yes?
What happens if you're missing?
There's some more genetic disorders that it's called chromosomal disorders. Or for example, Down syndrome, they have an extra number 21. They'll have 47 total. When we get to our genetics unit, we'll learn about something called Klinefelters, where they have 2 X's and a Y. They have 3 sex chromosomes instead of having 2. Turner syndrome is another one where they have they're missing an X, so it's like a woman who has just 1 X chromosome. She's instead of having 2. So we'll learn what the result of that is later. Let's talk about gene expression and where we're going to understanding DNA to RNA to amino acids, and those amino acids make up the traits, the measurable traits that we can see. That's called the central dogma of genetics, DNA to RNA to amino acids. You're going to hear us say it again and again and again. So just to recap, let's use the light panels. So let's say each one of those panels and the panels here are chromosomes. And we'll just use a little on switch, off switch to represent. This is just a model. It's not 100% accurate because during interphases, DNA chromosome or chromatin? Chromatin. So if I say those are chromosomes, so we're going to just factor that in. Okay, so you start out as one set of genes, right? And the lights are off. So imagine every one of those panels being your different chromosomes. If you add them all together, we call that your genome, the total genes that an organism has. Okay? So I'm going to turn the lights on for a second just to demonstrate. So here's a cell, and it's going to start dividing. And every one of those divisions is basically increasing the number of cells. But it hasn't started to specialize. So, like I said, just imagine it being a blank slate. So if you watch my hands here, this is now a ball of cells. That's how you work. And then, see my fingers going down? They start to go down, go down. Some of the cells start to tuck in and become the inner lining. Some become the outer lining, like your epidermis. We're going to learn about ectoderm, endoderm, mesoderm, stuff like that when we do animals, there's a part on embryonic development, which is going from one cell to a multicellular organism. So, now, let's just say, again, the lights are off, meaning there's no specific genes turning on for differentiation. There's genes turning on to, say, increase the number, but then how do you get from that one set of genes that mass produces itself through mitosis, growth and development, that's where genes start specializing. So let's say in one set of cells, those genes are turning on. All right? Versus up front, these genes in the same cell are off. But now let's flip it. Let's say another set of cells, these genes are on, but those ones are off. They're all the same genes, but not all the genes are on at the same time. About 95% of your DNA at any one particular point is not on. They're wrapped up. There's a physical and a chemical barrier. Remember, chromatin is DNA and protein, so you wrap the DNA around a thing called a histone, and you wrap it, Remember, you further condense it to make a chromosome. So all your genes are not on at the same time. If they did, you'd have massive problems. You'd produce too much protein, or you'd have a system shut down. Okay, so as you start developing, say, some of your cells are developing that form the epidermis. That means other cells are turning genes on. They're going to become the stomach cells. All right, and you have this as you got this cacophony of lights turning on, genes turning on. I'm not trying to cause you to have a seizure, but the lights going on and off and on. My whole point is that's showing a gene turning on, but not all the genes are on at one particular time. Does this make sense?
Yeah.
Yes, no, maybe. If not, okay. So what? No, I'm not going to keep turning the lights on. My point is, if you were to look at all your cells have the same genes, but not all your cells are expressing genes at the same time. The same genes. They're doing different genes when there's a need. All right?
Okay.